Good evening. On behalf of the Islesboro Forum Committee, welcome to the second program of the 2021 Islesboro Forum Speaker Series. I'm Gail Jackson and the moderator of today's program. This is our 29th year of presenting distinguished speakers who are experts in many different fields, presenting them to the island community. Last year, because of COVID, for the first time, we presented our programs live via Zoom webinar rather than in person. Many of you have been asking, how are we going to do it this year? We have 7 programs scheduled, one more than last year. The first 3 will be via Zoom. For the final 4, we will have the speakers in person at the community center, just like the old days. And as well, the program will be available to watch via Zoom. We hope to welcome as many of you as possible in person for the final 4 programs. The entire calendar of programs is listed at the website. Make note of this, islesboroforum.org. Before I introduce our speaker, I want to acknowledge and thank the 7 other committee members for their terrific support and ideas for this season. Their names are listed on the website. Also to be commended is our technical producer, Michael Hutcherson. Finally, please share your comments and speaker ideas for future seasons via our website. And while on the website, please consider making a donation. Our cost for 7 programs will be about $1,500, not counting the volunteer hours given by the committee. We hope you will want to support the speaker series with a donation. A quick reminder as to our format: once our speaker has completed his remarks, we will have time for question and answers. You should find the question and answer function on the bottom right of the Zoom webinar screen. Type in your question, hit the enter button, and the moderator will recognize you according to where you are in the queue. Now, it is my great pleasure and privilege to introduce tonight's speaker, Dr. David Van Essen, a world-leading expert on the science of brain mapping. Dr. Van Essen has accumulated a list of achievements that is admirable and impressive in the extreme. He obtained his undergraduate degree from Caltech, his Ph.D. from Harvard, joined the Caltech faculty in 1976, and was recruited to Washington University in St. Louis to chair the Department of Anatomy and Neurobiology for 20 years. His many leadership positions include president of the Society of Neuroscience and founding chair of the Organization for Human Brain Mapping. His awards are too numerous to list, but 2 that sum up the high regard in which he is held by fellow scientists are his election as a fellow of the American Association for the Advancement of Science and his membership in the National Academy of Sciences. Those organizations don't just hand out these distinctions on the street corners. He is in rarefied company. Our forum committee is enormously grateful that David accepted our invitation to speak, and I will now turn it over to him to take us inside our heads in a way that few others can. David, over to you.
Thank you, Gail, for that generous introduction and for the opportunity to speak to the Islesboro Forum about my scientific passions. You can see my screen, I hope. Okay, so let's jump right in. if you think of what's inside Gail's skull or mine or yours, we are all endowed with brains that are similar in broad strokes, but different in many details, that it can account for our amazing capabilities to cope with language, tool use, thinking, creative generation, and other things that make us very special. We know that our evolutionarily closest living relative is the chimpanzee, and they're actually intelligent creatures too. They have some language capabilities, can do a bit of tool use, but their brains are only a quarter the size of ours and are profoundly less capable in many respects. So with that in mind, let's put our heads around the following big questions. How does the human brain work to make us unique as a species, unique as individuals, gifted with amazingly diverse talents, and yet inflicted by devastating brain disorders that can strike at all ages and stages of the lifespan? I hope you'll agree with me that deciphering the brain in health and disease is truly a grand challenge for the 21st century and beyond if our species is both wise enough and lucky enough to survive. I want to focus tonight on the cerebral cortex, mainly in humans, but also in animal models, especially the macaque monkey. I love numbers about the brain, so let's look at a few. Each of us has about 3 pounds of squishy brain tissue inside our skull. It contains about 90 billion nerve cells or neurons. If we take a slice, what we call a coronal slice, midway through the brain and look at it, we see that there's a slab that has both gray matter on the outside and white matter internally. If we take a slice along the midline between the left and right hemispheres, we see a view that also shows the cerebral cortex to be the dominant structure from all perspectives. Cerebral cortex and the underlying white matter together constitute about 80% of the brain mass, and they're responsible for amazing diversity of functions, our perceptions, our control of movement, our cognitive capabilities for language and decision-making, and many other distinct functions. And yet it's done with only about 16 billion neurons, that is 20% of the grand total, but it also takes advantage of about 150 trillion synapses. These are the little communications packets that communicate from one nerve cell to another. In the underlying white matter, there's approximately 100,000 miles worth of axonal fibers connecting one brain region with another. In the pint-sized cerebellum that's at the rear of the brain and beneath the cerebral cortex, the cerebellum constitutes only about 10% of brain mass, but it has an amazing 80% of all the neurons. And it's very intimately involved with coordination of our movements and also our thought processes. The rest of the brain, besides the cerebral and cerebellar cortices, are what we call a collection of subcortical nuclei. They are less than 10% of brain mass and fewer than 1% of all the neurons, yet they're extremely important for what we call housekeeping functions, making sure we breathe, eat, and take care of our bodily daily functions. In the rest of today's presentation, I want to talk about cortical cartography, that is how we make maps of cerebral cortex and how we use these maps to explore individual variability as well as cortical development. I'll talk some about cortical connectivity in our animal model, the macaque monkey, and then shift over to the main agenda, which is the Human Connectome Project, and the way it's given us high quality images using magnetic resonance imaging of many different modalities to explore structure, function, and connectivity, and how that's led to a much improved 21st century map of human cerebral cortex. We'll talk a little bit about evolution and cortical expansion in the human lineage towards the end, and then we'll wrap it up and I'll be happy to take questions. So we've already noted that our brains are 4 times larger than that of the chimpanzee, which diverged about 6 million years ago, but the greatest amount of human cortical expansion occurred just in the last couple of million years. Our brains are 15 larger than the intensively studied animal model, the macaque monkey or rhesus monkey, 200 times larger than another widely studied primate, the marmoset monkey, and we're 3,000 times larger than the tiny mouse brain, a rodent species that is extremely intensively studied amongst the neuroscience community. So at a glance, you can see that the folds or convolutions of the cerebral cortex vary enormously from nothing in the mouse to varying degrees and patterns across species. And it's these folds that give rise to the need for cortical cartography of sophisticated map making. We want to put on our maps information about brain structure and brain function. One type of structural information, it relates to the cellular architecture or configuration of distribution of cells within the cortical gray matter ribbon. So on the upper left, you see at low power, a slab or slice of the human brain in which the purple staining shows the distribution of neuronal cell bodies. And at higher magnification on the right panel, you can see little tiny dots that are each the single cell body, and they're distributed throughout the cortical sheet, but more packed in, denser in some regions than others. That gives a layering pattern that changes from one region to another. And it's these subtle differences in layering and cellular distribution that the great German anatomist Brodmann a century ago used to identify 50 distinct cortical areas by his partitioning or parcellation scheme for cerebral cortex. Now, each nerve cell whose cell body is stained purple in the view there also has little branches in the nearby region. These are called dendritic arbors or branches, and they're essentially the receiving side, the input side for each neuron, which then decides whether to send information that is integrated into nerve impulses that travel along what's called the axon, a single thin fiber that goes long distances to contact other neurons on their dendritic arbors. And each of these contacts is called a synapse, and that's the mode of communication from one nerve cell to another. These axons, it turns out, are insulated by a fatty sheath that's called myelin that's wrapped around each axon, kind of like the insulation and the wiring for your electronic devices. And that myelin insulation turns out it can be conveniently stained by special staining agents that turn the white matter completely jet black in the middle region of this right side panel. But within the cortical ribbon, it's varying shades of gray at low power and at high power as the distribution of myelinated fibers changes from one brain region to another. And that type of information was used by a different group of German anatomists, Cecil and Otto Vogt, at the same institution as Brodmann, but a decade later, they used myelin staining and myeloarchitecture analysis to identify what they consider to be 200 distinct cortical areas. And this conundrum is that approximately 50 or 200 or somewhere in between is something that's been puzzling neuroscientists for a century, and we'll return to this later in my presentation. But you'll also notice that these classical maps that Brodmann and the Vogts provided are incomplete. You can't see into the interior, the 2/3 of the cortical sheet that's buried in sulci in these external brain drawings. And that's also true in the image you see on the lower right, which is that of a macaque monkey. Brodmann had a map for the macaque that identified approximately 40 distinct cytoarchitectonic areas. When I started studying the macaque in my research beginning about 45 years ago, I was frustrated by these brain drawings and their incompleteness, and I took a tip from the world of Earth cartography. As we all know, Earth cartographers have for centuries been making flat maps of the Earth's cortical surface. We can take the spherical Earth, make cuts in it, flatten it like a pancake, and also flatten out the mountains and valleys that are undulations or ripples in that surface to make it completely flat. We can display on those Earth maps physical features, geographic or other characteristics. We can also paint maps of the political subdivisions, countries, states, counties, all related to the distribution of the people that populate the Earth. And lots of information can go on these maps. So for the map of monkey or macaque cerebral cortex, when I started on this 45 years ago, we didn't have computers powerful enough to get the job done. So I literally used pencil and tracing paper to make these charming but quaint maps of the macaque cortical sheet. And it had cuts in it to smooth out the curvature, and it showed in the gray regions the buried cortex within these deep sulcal folds. We also painted on these maps more colorful representations, in this case of 32 distinct cortical areas associated with processing visual information, because the macaque, like us, is a highly visual creature, as well as dozens of other cortical areas associated with other functions. And these cortical parcellations into different cortical areas are based on information that Dan Fellman and I drew from the literature relating to cortical function and architecture and connectivity and topography, different kinds of information that gave us a basis for identifying cortical areas. And a few of these areas, such as the primary visual area V1 on the left and the little reddish area MT on the right, were particularly well identifiable by any or all of these criteria. They were our poster child areas because they were well defined and also well understood. V1 is a sheet-like area that in which each patch of the cortical sheet analyzes each patch of our external visual world for features of interest, edges and contours and colors that are in and constitute our visual images. Area MT, on the other hand, is specialized for analyzing a specific type of information, whether things are moving, and if so, what direction are they moving? So this gives you a hint, but when we look at these other cortical areas, the story gets a bit murkier, in large part because these other areas are not nearly as well defined. We draw sharp borders, but the evidence behind that is fuzzy and often controversial. And to make a long story short, cortical parcellation was and still is a very hard problem, but it can benefit from new methods, and particularly for the human cortical sheet that we'll talk about shortly, magnetic resonance imaging has turned out to be critically important. That came into play in the 1990s, and it allowed us to go from viewing just slabs of the postmortem brain, like you see on the left and had seen in an earlier slide, to MRI images that you can get for your own brain if you're willing to lie quietly in an MRI scanner and have an image like you see in the upper right, which shows the cortical gray and white matter from an MR scan. This is one of many types of structural images about the brain configuration that can be viewed with MRI scans. We can also get information about brain function using what we call functional MRI. And we'll see soon how we can pull together information about function, architecture, connectivity, and topography, the same four categories of information, but now extract it using MRI to get our maps of human cerebral cortex. So let's first start with structural MRI. And if we take a high resolution structural MRI of a healthy human, you can see in this movie, scrolling back and forth, the blue contours and the greenish contours outline the inner and exterior limits of the cortical sheet. And we can use that information to make three-dimensional brain models, which we can spin around to get a view from different perspectives. And we can also physically or computationally inflate the cortical sheet and make a smooth map where the darker regions represent regions that were deeper in the cortical sulci in the original configuration. If we do this for many individuals, we can see in these four exemplar individuals in the upper left here that each individual's cortical folding pattern looks different than the others, much as human fingerprints are not identical across individual subjects. If we smooth out each of these individual cortical right hemispheres and look at the pattern of what we call these sulcal depth maps, you can see that in some regions, there is a consistent pattern, such as this fold that we call the central sulcus. But in most other regions, the folding pattern is dramatically different from one individual to another. Remarkably, that's true even for identical twins. So lo and behold, the upper pair is from one pair of identical or monozygotic twins. The lower pair is from another pair of identical twins. And even though they received identical genetic instructions during development, the folding pattern is nearly as different in identical twins as it is across unrelated individuals. There is, in fact, some heritability, but that's not what strikes you in the face when you look at these patterns. It also turns out that the cortical areas that we heard about in monkeys and a little bit in humans, we'll hear more later, but those areas that are well defined, such as the primary visual cortex shown in red in these three individuals, these areas vary in size by 2 to 3-fold from one individual to the next. You may have a small area V1, but a larger area of MT and so on. And the boundaries of these areas vary in relation to these cortical folds. So we're going to touch on 2 additional questions relating to the folding patterns. One is, why is the cortex folded? And then the answer to that, I think, is simple. It's in order to take a large sheet-like structure. Your right hemisphere is about the surface area of a 13-inch pizza, and so is your left hemisphere. To get those pizza-like domains fit into the skull, they need to be scrunched or crumpled up, much like you'd crumple a newspaper to get it inside a small jar. But then the question is, physically, how during development, what makes the cortex follow that and become folded? Well, we can look at that issue in humans and note, first of all, that we know when cortical folding occurs. At the beginning of the 3rd trimester of human gestation, the cortex is actually quite smooth. There's only a few small folds. 3 months later, near term equivalent, just before the baby pops out, the cortical sheet is almost as convoluted in the near term equivalent baby as it is in the adult, even though the size is 1/3 that of the adult. When I thought about this 25 years ago, Tom Coogan, a postdoc in my lab, and I had just found that the connections between primary visual area V1 that you already heard about and neighboring visual area V2, that those connections are formed within the white matter, V1 connecting to V2 and vice versa, right around the time that a fold, what we call a gyral fold, appears between them. And I thought to myself, well, maybe that's not an accident or purely coincident. Maybe in my light bulb moment, there's a cause and effect here, and maybe mechanical tension along these literally millions of axons connecting V1 and V2, if they pull together, would bring strongly connected regions of gray matter closer together, forming this gyral fold in between. Whereas an inward or sulcal fold, like you see I'm pointing to here, could be the outcome of a losing battle where there weren't enough axons or they weren't pulling strong enough to keep them from getting pulled farther apart. So this one hypothesis of tension-based cortical folding can account for both the consistency that we see in some regions and also for the variability across individuals and across species in other brain regions. It is, in fact, an attractive hypothesis, but it's not proven, and it's likely to be just one of several cooperative or coordinating mechanisms. But the bottom line is the cortex gets folded, and in healthy individuals, it looks fine at birth. But abnormal cortical folding can occur in a variety of brain disorders. There's one particularly called Williams syndrome, which is characteristics of pattern of strengths and weaknesses in individuals who have this, and the cortex is folded abnormally. I'm going to illustrate just briefly another example of what happens sometimes during premature, very premature infants. They're at great risk for autism and ADHD and language disorders. And in contrast to healthy term-born infants, some infants born very prematurely, like at 26 to 30 weeks, have brains that are abnormally folded when they're tested at term equivalent. So here in these four examples are ranging from moderate to severe folding abnormalities, concentrated mainly in the temporal lobe, which might account for example, the high incidence of language deficits. So this just illustrates the importance of thinking about imaging brain structure and function to aid in the diagnosis and hopefully eventual treatment of different brain disorders. Let's return to the issue of cortical connectivity in animal models. I showed you previously a schematic of neurons, cell bodies, axons, and dendrites and synapses. We can use this schematic to make the point that in laboratory animals, it's possible to make injections of chemical tracers into a little patch of cortex in one area or another. And for some tracers, the tracer molecules are carried in what we call the anterograde direction from the cell bodies to the axonal terminations and synapses to reveal one direction of connectivity. Other tracers used in other animals can be used to identify the tracers that get picked up from the synapses and are carried along the axons in the reverse or retrograde direction and are found by their location in the cell bodies. So using these methods, many studies were carried out in the 1970s and 80s and 90s. And Dan Fellum and I analyzed many such studies published in the literature, and were able to report evidence for literally hundreds of distinct area-to-area pathways that are summarized and schematized in this essentially subway chart diagram of the cortical visual area network. And we hypothesize that they are configured in a hierarchy that goes from the primary visual cortex at the lowest V1 to V2 to intermediate and progressively higher order visual areas that carry out more advanced levels of visual processing. We also took the same information pertaining to hundreds of pathways and laid out the connectivity information in a matrix in which each row shows the outputs of each cortical area, and each column shows the inputs to each cortical area. So an X marks a spot when there is a pathway or projection from one area seen in the row to another target area seen in the column. You can't see their names and serial numbers, but you get the sense of it, I hope. So this is what we called a connectivity matrix at the time. Now in modern lingo, we'd call it a parcellated connectome. And the idea of a connectome has become very important and very exciting, because in the ensuing several decades, there's been an absolute explosion of rich and very accurate information about brain connectivity. And it leads us to the aspiration of describing the brain connectome as a comprehensive map of neuronal connections. But we're forced to put the comprehensive in quotes that we mean very strongly, because with the current technology, our story is very incomplete. There is a wonderful world of what we call the microconnectome that uses images and information from very high resolution electron microscopic images to visualize and reconstruct the full three-dimensional configuration of every neuron, cell body, axon, dendrite, and synapse in its entirety, but only over a limited range extent of a volume that's about a millimeter on a side. That's less than a tenth of an inch in extent. There's another world of what we call the mesoconnectome that uses the tracers, the anterograde and retrograde tracers that I illustrated previously, to look at local and long distance connections and build up a picture of three-dimensional connectivity, but it requires information obtained from many animals with just a few tracers you studied in each animal and a few of the many pathways, but it gives an increasingly rich portrayal. The last and the one I'll talk most about is the so-called macroconnectome world that uses MRI or other whole brain imaging techniques to identify pathways connecting long distance regions in the human brain with complete coverage of the brain, but a resolution that has the individual volume elements, or what we call voxels, only a millimeter or two on a side. That is the same size as the full extent of a microconnectome reconstruction nowadays. And each of those voxels contains literally hundreds of thousands of neurons, and that's the limit of our macroconnectome resolution with current technology today. So with that approach, the Human Connectome Project has studied the macroconnectome in many healthy young adults, and I'll illustrate what we get for representative different structural and functional and connectivity informing modalities, and how we use that to get an improved cortical map. So I had the tremendously good fortune to serve as the, and privilege to serve as the principal investigator for the Human Connectome Project that started in 2010. And over the ensuing 6 years, we got high quality data from 1,100 healthy participants scanned using an improved MRI scanner. And we got 4 hours of imaging data per participant, plus extensively acquired behavioral and demographic data, as well as blood samples for genotyping. And we took that information and analyzed it very thoroughly and very carefully using much improved methods for aligning across individual subjects and improved visualization methods and other advanced analyses. We then shared the data, not just in a scientific publication or two, but the actual data themselves with minimal processing or extensive processing, whatever people wanted. We gave them the data, the methods, and the tools, and shared more than 25 petabytes worth of data. And that's 25 million gigabytes, and a gigabyte's a hefty amount of data, as I'm sure you appreciate. And that data has been distributed to nearly 20,000 scientists around the world, who in the ensuing years have generated more than 1,400 scientific publications that make use of HCP data, and they're continuing to use it extensively, and that number of publications grows day by day. So to illustrate the types of information that emerged from this analysis, you see here a horizontal slice through a T1 weighted image, similar to one you'd seen before. But Matt Glasser, an exceptionally talented MD PhD student in my lab, had noticed that in the gray matter ribbon, some regions of the gray matter in this T1 weighted image are a little brighter than average, and other regions are a little darker than average. He also noticed that in another type of scan that we acquired for the Connectome Project, a so-called T2 weighted image, the contrast is the opposite. So now in the T2 weighted image, the white matter is actually dark, the cerebral spinal fluid is bright, and in the gray matter ribbon, the region that had been brighter in T1 weighted is now darker, and the region that had been darker is now brighter. So by combining these two types of imaging modality from structural imaging, and taking the ratio and then colorizing each little voxel and mapping it to the cortical surface, Matt and I were able to show that the regions where the ratio is high and colored red or yellow correspond to the regions of heavy myelination within the gray matter ribbon that had been identified by the classical anatomists that I showed you earlier. And the regions that are lightly myelinated are in these indigo patterns scattered throughout much of the cerebral cortex. When we do this and average across hundreds of subjects, we get very crisp and very informative, what we call myelin maps that are one type of information we use for identifying cortical areas and their boundaries where the sharp transitions in myelin content are seen. Another type of information is what we call a cortical thickness map, where regions that are thin, kind of like a thin crust pizza, versus regions that are thick, kind of like a thick crust pizza, are distributed in characteristic ways. And we also wanted to take information about other brain regions that are needed more information to properly parcelate or subdivide, using what we call functional MRI. And Deanna Barch, our colleague, designed 7 different tasks that were acquired in an hour's imaging period in the scanner. One of the tasks was just a language task, listening to stories and answering questions about them. Another task that took only a few minutes was a social cognition task, looking at these 2 movies. And on the right, you just see triangles wandering randomly and bouncing across the walls, whereas in the left, you see the triangles dancing and hugging and kissing, or it gives you that impression because of your social cognition capabilities. A third task is what we call working memory. Is the image you're currently viewing the same or different than the one you saw 2 images back in the sequence? And we'll portray these maps on a model that shows the left and right hemispheres from the lateral side on top and the medial side on the bottom. And if we take the story task, listening to stories activates in these red and yellow patches a strip of what we call the temporal lobe, and another strip plus a patch in the frontal lobe in both the left and right hemispheres. Whereas the next task, the social cognition task, activates a partially overlapping but largely different patch or set of regions, particularly in the occipital lobe shown by the blue areas. And the third exemplar task, the working memory task, trying to remember what you've just seen, activates mainly regions of the frontal lobe and also of the parietal lobe shown in the back of the brain. So that's task fMRI. All of our participants also spent 4 15-minute sessions, a total of 1 hour, doing nothing in the scanner, just thinking about whatever they felt like, but told to stay awake and keep their eyes open. And some of them were thinking about what's for dinner, and the others are thinking about who won the ball game, and others are thinking about that book they're reading, and so on. And during these free-form thinking periods, we analyzed the data and took the functional MRI signal that's shown in red for one subject, fluctuating up and down, and for another, the same subject but a different brain location, location 2, this fMRI signal fluctuates in a different way. And sometimes when we compared locations, we found the fluctuations were in sync with one another. They were highly correlated. In other cases, they were uncorrelated or even anti-correlated. And we took all possible pairs and generated what we call a dense connectome, literally 30,000 by 30,000 possible combinations, forming a functional connectivity matrix that we could then interrogate and say, what is the pattern of regions that are correlated strongly with this one location in parietal cortex? And those show up in red and yellow in different regions on the same and opposite hemispheres. Another example is correlation with this region of the central sulcus has a very different functional connectivity map. So we take this information of the different modalities and put them together and try to get a much improved map compared with the classical Brodmann and Vogt cytoarchitectonic and myeloarchitectonic maps. And Matt Glaster and I and our colleagues in the Connectome Consortium spent an intense effort in 2015 and 16 to analyze these data. And one example is this little tiny area outlined in black on this myelin map on the left. We call this area 55B, that is lightly myelinated. It responds very strongly when listening to stories in the second panel. And on the right panels, it is highly correlated with a region associated with language in the temporal lobe and anti-correlated with a region associated with eye movements that correlates instead with regions, patches on either side. And this identified this one area consistently using these multiple types of information. And when we applied this across the entire cortical sheet for both hemispheres, we came up with the map you see here, a map of cortical areas that identifies 180 cortical areas in the left hemisphere, a matching set of 180 areas in the right hemisphere. The green ones are associated with touch or somatic sensation, the blue with vision, the red with hearing, and the bright and dark with various types of cognitive processing. We gave each of these areas a name or serial number. 83 of them, fewer than half, matched well with previously reported or published areas. 97 we call newly identified because they were not previously reported, although some of them involve splitting of existing areas. And very importantly, nearly all of these areas could be identified not only in the group average you see here, but also in individual subjects. And these areas vary across subjects in their size, shape, and precise location. And each of them has a distinct signature of functional and connectivity characteristics. So this is what we've gotten highlights from the young adult HCP project. I'll just mention briefly that there are many other large-scale projects similar in spirit, motivated by this HC style mantra of getting high quality data in large amounts from lots of subjects and analyzing in the best possible ways, and then sharing the data freely. There's one set of studies looking at older adults, the aging project, another looking at younger children, an exciting project that's looking at 10,000 or 12,000 subjects, studying them over 10 years. It will be an enormously valuable resource for looking at development during the teen years. There's a baby connectome project, a developing human connectome project, and more than a dozen projects that are looking at specific brain disorders or diseases related to dementia, depression, psychosis, and other disorders. In the final moments, I'll just touch upon some issues of evolution and cortical expansion. You've already seen this myelin map earlier. I'll just point out that the red and yellow regions that are heavily myelinated constitute only a small fraction of the human cerebral cortex. Most of it are the blue and indigo regions that we associate with higher cognitive functions of one sort or another. And yet, if we compare to the chimpanzee, recall that the chimpanzee brain is much smaller, but in addition, the regions that are blue and indigo that we are associate with cognitive functions are a much smaller fraction in the chimpanzee than in the humans. And in the macaque monkey, it's smaller still, and it's a smaller fraction that is cognitive. And lastly, the little marmoset has its tiny brain filled with small patches of cognitive function. So we think this gives us a heads up on explaining, just in terms of the portions of cortex devoted to different brain functions, why our cognitive capabilities involves vastly more cortical turf, and we can compare it to, for example, the macaque monkey, and see that the regions of areal expansion in the human lineage compared to the macaque lineage during evolution is concentrated in these regions of lightly myelinated cortex specialized for many cognitive functions. So to sum up, cortical parcellations involve closer to what the votes estimated of about 180 areas per hemisphere in humans. Fewer than that, but still more than 100 in monkeys, but the jury's still out on that. The principles of cortical connectivity should reveal highly distributed pathways, but forming a hierarchical organization, many levels of processing, and the principles of wiring and circuitry that are very different from human brains versus digital computers. The field of connectomics at micro, meso, and macro levels is providing exciting new insights and discoveries, but we're long ways from having a complete human connectome. May never achieve it. but animal studies are vital for helping interpret human maps and connexomes, and high quality HCP style studies of development, aging, and diseases are going to improve our understanding of the brain in health and disease. So just as classical maps of the Earth's surface and the brain have changed dramatically over the years and benefited by improved technology, and up to satellite images for the world and MRI images for the human brain, and Google Earth transforming the way we navigate the world. And we hope that our new tools for exploring connectomics will take us to the equivalent level of sophistication that Google Maps provide for navigating the world. So I just want to say briefly, thanks to a tremendous collection of individuals in my lab and in our HCP consortium, and I'd be very happy to answer any questions.
David, thank you very, very much. I'm somewhat overwhelmed by the complexity of what you have been working on. And it seems as if as much progress as you've made in improving the cartography, if you will, you feel as if you've barely scratched the surface, that there are many, many additional areas that you and your colleagues around the world are going to be focusing on in the years ahead. I'm fascinated. Yeah.
That's true, and it makes me hearken back to when I was a graduate student and a postdoctoral fellow at Harvard University, and my mentors, David Hubel and Thorsten Wiesel, very shortly thereafter won the Nobel Prize for their pioneering work. And I was under the severe illusion that they had discovered most of what was exciting to be learned about the brain. And I was wondering, would there be enough left for me and my colleagues to discover in the years ahead? And the reason I mention it is because it dawned on me a few years later that the more we learn about the brain, the more we learn there is left to discover. And it's like an endless set of Russian dolls, but inside each doll is a smaller doll with higher resolution, but vastly more left to be discovered. And I think it will go on as long as the human species is alive and well to explore in deeper and more exciting ways these true mysteries of brain function.
David, we have some questions. I'm going to throw one out to you from myself, and then we'll go to the others. Does your work lead you to conclude that our fears and/or expectations about what artificial intelligence can do are perhaps exaggerated or overblown because the complexity of the brain seems to be just so far beyond what might be produced through computer-generated artificial intelligence?
Well, artificial intelligence did struggle for many decades to pass muster as a serious competitor to the human brain. But now computers can easily beat humans at chess or Go or any of the games that we contrive to challenge our own intellects. And more importantly, computers that emulate brain architecture in increasingly serious and sophisticated ways are proving to be more and more powerful. So there's a whole domain of computer analysis and artificial intelligence called deep neural networks. And they haven't by any means achieved the full richness of the human brain as we understand it, but they are approximating some of the important principles of our brain's ability to perceive and analyze vision and complex images, or to analyze other important and challenging data resources. And they are getting much better. Whether it the science fiction movies that show the computers taking over the world, for better or worse, will materialize. I kind of doubt it, but it's not out of the question. That it is certainly to be expected that the improvements in computer artificial intelligence is likely to accelerate rather than reaching a plateau. So it's anybody's guess where that will end.
All right, thank you for that perspective. Now we have a question from a physician, and she is beautifully precise. She says, "What's next?" Question mark.
Well, there are literally 30,000 neuroscientists in the Society for Neuroscience, and probably more than 100,000 neuroscientists practicing their skills on probing many aspects of brain function and structure and development. And so the number of discoveries and exciting insights continues to accelerate. One of the big what's next is, when are we going to cure some of these horrendous brain diseases and disorders? You know, cancer, after decades of struggling, is finally making dramatic advances in curing a growing fraction of the complex array of diseases, not just a single disease that we now realize cancer to be. And that's as a result of decades of investment in basic science of cell biology that has given us the information and tools to tackle cancer. For the brain, there have been decades of investment in studying Alzheimer's disease and autism and many others, and they haven't given way as yet to comparable success. And I think that's fundamentally because it's just a profoundly greater challenge, and we're not as far along as we need to be in understanding the basic circuit abnormalities. Despite the progress I've described, we've got a long ways to go, and the tools for advancing will come along. But I'm cautiously optimistic that in the coming decades, these investments in basic and clinical research will pay off, but it's hard to predict which ones. But there are powerful tools that will bring to bear on that. And so there's reason to be hopeful, but I stress the importance of being patient, even as people are desperate to see progress in these arenas.
Thank you for that. Now, in a moment, we're going to try to bring in an old professional colleague of yours, Barbara Talamo. We're trying to unmute her mic, but until we are sure that we've got her set up to ask her question directly, I'm going to pose another question. This comes from an economics professor. Does anything you are learning give us ideas of what we should be doing to maintain our brain health as we age?
There is certainly a lot of research on that. First of all, I won't pretend to be an expert on it, but I think the fundamental advice I would give is probably consistent with what my mother and many of your parents would have said, is stay active. Activity improves physical health, and there's some evidence and a lot of intuition to support the idea that activity, challenging your brain, helps keep it working well. And so that would be the most important thing I would stress, whether you choose to play games with your grandchildren or what, it's the best exercise is stimulating your brain in diverse and rich ways. Can I respond to one of the interesting questions I see here, Gail? Once you identify a potential connectome pathway, how do you test its function? That's a wonderful question, and it's a really hot area in brain research. It turns out there are many ways and a growing number of ways to perturb brain circuits. And some of these involve studies in mice and powerful techniques for being able to turn off or turn on individual neurons or small clusters of neurons in a behaving animal and say, what can it suddenly not do or not do as well as it did when you silence a neuron or a group of neurons, or tickle them and make them more active than would have been otherwise. And all sorts of cool stuff is being revealed by this, relating to how we perceive, how we control our movements, and also very importantly, some of these are having a real impact in what we call brain-computer interfaces. So it's possible in paralyzed individuals to listen to the brain and turn those signals into robotic activity. So to get up in the morning and for a paralyzed individual, say, I want a cup of coffee. Right now, the tools are able to let a patient pick up a cup and drink from it. And these tools are getting better because of insights we're learning about how the brain computes and signals, and the idea that a paralyzed individual would be able to make the coffee as well as pancakes, as well as eat them. These are very much in a realistic stretch. Making blind people see is also an, it's not there yet, but the capabilities are on the event horizon. And I'm optimistic that those kinds of advances will be impactful in a small number of years.
Well, that's thrilling to hear. Now, I think we have Barbara Telemo. For those who don't know her, I believe she was chair of the department, counterpart department at Tufts that David was chair of at Washington University. So over to you, Barbara, if you can be brief and concise. We have a number of other questions too.
I'll try. Hello, David.
Hi,
Can
Barbara.
you hear me?
Yes, I can.
Beautiful work. And I was wondering whether you have an idea about how an approach to consciousness might be attacked through these techniques. I was thinking of a book called Into the Gray Zone, where the author writes about experiments with people who are not only in locked-in syndrome, they can't communicate at all externally, but he's determined by functional MRI that they are conscious and aware of what's being said to them. And is there any way to get at that using some of these techniques?
The answer is very likely that will be possible. Functional MRI is one way, and I'm not expert on those studies. I'm aware of them. I think it's a bit controversial as to how real or convincing and compelling it is, but whether or not the answer is convincingly known now, I think fMRI is one of the tools that, if properly used, will likely give clearer answers to whether some of these locked-in patients truly have conscious capabilities. The other methods I referred to relating to brain-computer interface, there are increasingly sophisticated and safe to use tools for putting electrodes onto the brain inside the skull, or even into the brain, stably and safely listening to large numbers of individual neurons. And those are going to allow deeper and more compelling, convincing assessments of brain signals introduced by subjects thinking about where they want to move, or what they want to write, or what they want to say, or other capabilities. So I think there's much that can be anticipated on that front. With regard to the issue of consciousness, Christoph Koch and others are fascinated by this, and some have speculated that a particular structure called the claustrum just underneath the cortical sheet is an intriguing anatomical structure that might be specially related to consciousness. Others are more skeptical of that and are able to explore higher regions of the visual hierarchy that I showed, and in those higher levels to probe individual neurons and ask, for example, is there or is there not a set of grandmother cells, that is, neurons that are active only when you think of or see your grandmother, and when they fire, that gives you the percept of your grandmother, which would be getting close to the component of consciousness. The answer is not out yet, at least to my awareness or satisfaction. But again, the tools are getting much better, faster enough that we can envision intriguing answers to such intriguing questions in the coming decade, I would say.
Thank you, David. I'm going to extend our time ever so briefly to slip in this last question on synapses. Can you see it in the chat? Kara Masters asks you to elaborate on the number of synapses, their space, are there multiple varieties? How is the synaptic space? Is it thick, liquid, pH, acid, charged electrically? What are the other characteristics of the synaptic space? And if you can give us a concise answer to probably a very complex question, that would be wonderful.
So a synapse is a tiny little structure, less than 1 micron, 1/1000th of a millimeter on a side. That's why you can get 150 trillion inside your skull. And there are many varieties. One basic category, some excite and some suppress or inhibit. But beyond that, there are hundreds, if not thousands, of variations, of subvarieties that differ in their molecular machinery. And so when you say what is in that space, thousands of specialized molecules mediating the signaling from one neuron to another. And these are the substrate of learning and what we call plasticity. The adjustments in synaptic strength are fundamental to how we learn or how we forget. And so there's an immense amount of work and lots of exciting progress just in the realm of exploration of synaptic machinery and its role in behavior that I haven't had time to more than scratch the surface to a tiny extent.
Well, I think it's clear, David, that you're never going to be able to retire. You've got a lot ahead of you. I'm going to have to bring it to a close now. I want to thank you again for taking the time to put together this fascinating talk, which gives us somewhat of an idea of the complexity of the organ that you're dealing with, and makes us very grateful that people like you who are so persistent and who have such curiosity continue to work on it with such energy. So thank you so very much. And if you'll just permit me, I'll say a few words about next week's program. So turning to next week, on July 25th, we will present Dr. Myron Cohen, who is director of the Institute of Global Public Health at the University of North Carolina. His talk is entitled COVID-19, Past, Present, and Future. He is recognized globally for his expertise on a subject that none of us go a day without thinking about, but we crave more information, and he will be able to help provide some of that. So please join us next week and join me in thanking Dr. David Van Essen once again for his wonderful talk today. Over and out.